بسم الله والحمد لله والصلاة والسلام على رسول الله. اللهم علمنا ما ينفعنا وانفعنا بما علمتنا وزدنا علما وعملا صالحا متقبلا يا رب العالمين وافتح علينا فتوح العارفين. Hello my friends, we have learned previously about the structure of a DNA and how a DNA can be replicated. But in this video, we have to learn the transcription and translation of a DNA. In other words, we have to know how a DNA is transmitted into a protein. Let us learn together. But before we go deep in the process of the protein synthesis, we have to know the importance of these proteins. So proteins are important in the growth of children, teens, and pregnant women. Also, they maintain proper pH and food balance. Third, they are responsible for many traits in our body. As an example, the color of our eyes. We inherit the color of our eyes from our parents. But have you ever asked yourself, what's the cause behind having different eye colors or the color of the eye itself? In this video, we will learn the cause for that. And also, proteins play a major role in transport, in structure of the cells, and also as enzymes. Also, as for the answer for our question that it is the cause behind having different eye color is short as such. Firstly, the pigment that it is in our eyes, that it is the eye color also, is due to a gene, which is a portion of our DNA, that encodes for a certain protein that gives the eye its current color. What are the steps that they are involved in a protein synthesis? Firstly, the gene is encoded into RNA. This RNA is encoded into a protein through a gene expression process. This gene expression process is divided into two processes. The first one is DNA transcription, and the other is DNA translation. We have to know quite a bit differences and similarities between both of them so we can differentiate. Firstly, the transcription and translation processes are common in the number of their stages. We have three stages for transcription and translation: initiation, elongation, and termination, but of course with different characters. As for the differences, transcription occurs in the nucleus, while translation occurs in the cytoplasm. Transcription also uses the DNA as a template to make the mRNA, while mRNA is used to make a protein in translation. So let us learn first about the transcription process. The first stage is initiation, and it's referred to as the binding of the RNA polymerase enzyme into a sequence of the DNA that it is referred to as the promoter. This promoter is a DNA sequence that causes the RNA polymerase to bind to make the RNA. This is the RNA that is present in this figure. But of course, we have further more steps to develop an RNA. It's not only developed due to the binding of the RNA polymerase into the sequence, of course, okay? So also, as it binds, it causes the DNA double helix to open and unwind. So as for the next stage of transcription is known as the elongation phase. It starts by the sliding down of the RNA polymerase enzyme down through the template strand that it is located in the 3', 5' direction, and not through the non-template strand that it is located in the 5', 3' direction. As you are seeing in here, this is the RNA that it is being synthesized in the 5', 3' direction, of course opposite to the direction of the template strand. But an important issue we have to focus on is that in the RNA, we don't have the T nitrogenous base. We have the U nitrogenous base, the complementary nitrogenous base for A. What's meant by that? Let us look at this adjacent figure. G is the complementary nitrogenous base of C, and C in the RNA is the complementary nitrogenous base of G in the DNA. A is the complementary nitrogenous base of T in the DNA. But in case we have A nitrogenous base in the DNA, The complementary nitrogenous base for it is not T, it is U. That's what's meant by U being the complementary nitrogenous base for A instead of T in the RNA. So finally, how this transcription is terminated? It's terminated by the RNA polymerase being binded to a terminator sequence of the DNA, saying that it is fully synthesizing the RNA strand that it is needed to be used in the synthesis of proteins. So this is the mRNA that it is obtained from the transcription process that it is discussed previously. As for the differences and similarities between RNA and DNA should be known in order to avoid confusion between both. So RNA and DNAs are both nucleic acids, but we have in RNA a ribose sugar. Instead, in the DNA, we have a deoxyribose sugar that it is discussed previously, remember? And also, we have single-stranded RNA in comparison with double-stranded DNA. As for the nitrogenous bases present, A, C, G, and U are present in the RNA, while A, C, G, and T are present in the DNA. We have talked in the previous slide that the complementary nitrogenous base of A is U and not T in the RNA. As the transcription process is ended, we obtain this immature mRNA that contains the exons that they are numbered from 1 till 4 and are known as the coding regions of this gene. And also the introns that they are known as the non-coding sections or the non-coding regions. To be used later in translation, we have to remove these introns, so they are spliced out by an enzyme that is known as the spliceosome enzyme by a process known as intron splicing to obtain only the coding regions of exons only and to obtain this mature mRNA that should be used later in translation. So how is this mRNA translated into a protein? The first step of translation process is initiation. This initiation stage corresponds for the binding of the mRNA into the small ribosomal subunit. Second is that the binding of this initiator tRNA into the start codon of the mRNA. We don't have to forget that the direction of the mRNA is in a 5', 3' direction. But wait, what's a codon? A codon actually consists of 3 nitrogenous bases. So as you are seeing in here, we have A, U, and G that serves as a codon. So this initiator tRNA binds to this start codon through an anticodon in a complementary way. So U is binded to A, and A is the complementary nitrogenous base for U. Also, C is the complementary nitrogenous base for G. This allows the transmission of the initiator amino acid methionine or MET through this initiator tRNA to start the translation process. The third step is the binding of the large ribosomal subunit. So it forms the translation complex and initiation is complete. As for the elongation process, a tRNA again comes up with a specific amino acid corresponding to this specific codon on the mRNA. So it binds first to the A site, which is called the amino acetyl site, and it's the first binding site of the tRNA. Then it binds to this P site, which is called the polypeptide holding chain site that holds this amino acid chain or its amino acid sequence, which are the monomers for this polymer that will be formed, which is the protein. These amino acids are linked together by a peptide bond. And then this tRNA is released from the ribosome through an E site, which is called the exit site. Then how is this translation terminated? As you can see in this slide or in this figure, we have a release factor. This release factor is binded to the A site at a stop codon. By the way, we have three types of stop codon that we will be talking about in the upcoming slide. Thus, this synthesized protein is dissociated or is released from the tRNA in the P site, and thus the complex is dissociated. Also, we forgot to mention an important RNA, which is the rRNA, that serve a critical role in forming catalytic sites of translation for mRNA and also is a major constituent of ribosomes. So here I can give you an example that we can predict by it, or we can know what's the polypeptide from this mRNA. So as I have said that three nitrogenous bases correspond for a codon, and we have only one start codon, that it is the AUG, and we have three stop codons which are UGA, UAA, and UAG. Also, an important information I want you to know is that maybe we have more than one codon that can code for one amino acid. As an example here, CGU and CGC, CGA and CGG codes for ARG amino acid. For example, CAU and CAC codes for histidine or his amino acid. So as I have said, we might have more than one codon that can code for a certain specific amino acid. Now, how we can know the amino acids? So as an example in here, the mRNA, the first codon is AUG. How we can look for this in the table? The first letter of the mRNA codon is A, so this is A. The second letter is U. This is the second letter, and this is U. The third letter is G, so I look for this third letter, and it is G. So firstly, A and U. So this is the box, okay? The first letter is A, the second is U. So A and U, this is the box. But in order to know which amino acid, we have to look for the third letter, which is the G. Here is the G, okay? So AUG corresponds for MET or methionine, which is the initiator amino acid or the starting amino acid for this start codon. And also we have only one start codon, as I have stated earlier. So a second example is, let's take this codon, UCG. The first letter is U, so this is U. The second is C. This is the second letter, C, so this is the box. The third letter is G, so UCG, UCG. So, okay, UCG. This is the codon. Okay, UCG, which corresponds for ser or serine amino acid. Another example is the UAA. Let's take UAA codon. This. The first letter is U also, the second letter is A, and the third letter is A. So we conclude here that it is a stop codon. So this is the end of translation process. So as I have said earlier, that this protein, once it is dissociated entirely from this complex or from the translation complex consisting of the mRNA, the small ribosomal subunit, the large ribosomal subunit, the tRNA, it should be modified and folded in order to give the right protein that is responsible for making or giving a normal function in our body. And here we have ended our video and we have reached its end. So I hope if you have any questions to state it down in the comments, and do not forget to subscribe, like, and share my video. Thank you for your watching, and bye bye.